Hello students, in the previous classes we have discussed about the fertilization process or the pollination, all the events which are involved in the sexual reproduction of flowering plant. This is the last topic, which is the development of embryo. After fertilization, embryo is formed, zygote is formed, and it results in the formation of an embryo. The process of formation of embryo is quite different in both the dicots and monocots. Today we will discuss about the developmental process and a few terms related to the reproduction, if like parthenocarpy, polyembryony, apomixis, then fruit formation and seed. So let's start with the development of dicots. In dicot, as you can see, the diagram appears to be complicated, but actually the process is very simple and easy. So just you have to, what you can do to learn the process is just repeat what I am saying. Just repeat once or twice and you can easily understand the process for the development of dicot or monocot embryo. Let's start with this one, dicot. In this, it is the zygote. First, A is the zygote. In this, the dot, this portion represents the embryo, and this one is the endosperm. Endosperm, which is the protective covering or the nutritive layer, nutrition, which provides nutrition to the developing embryo. After this, the first division is a transverse division, which results in the formation of apical cell and basal cell. Apical and CA is for apical cell, CB is for basal cell. After that, the apical cell, and there is one more transverse division, and the basal cell, which is in close contact with the micropyle. Basal cell is in close contact, close proximity with the micropyle. CI is for cell closer to the integument, CM for the micropyle, close to the cell which is close to the micropyle. Then here I have skipped one stage before this, which results in ultimately the formation of these two basal cell dividing into two, into integumental and micropylar, and the CA undergoes a longitudinal division. Before this, there was a transverse division, but the apical cell undergoes a longitudinal division and form this type of structure, which is the C. After two cells in the apical cell again undergoes the transverse. First is the longitudinal and then a transverse. This results in a formation of a quadrant, this type of quadrant. This quadrant is formed, and these cells, the basal cells, they form the suspensor. Next to this is the quadrant undergoes again transverse division like this. So this transverse division results in the formation of 8 cells, which is octant. So the structure, which is this type of structure, is formed with this basal cell and the suspensor and the octant. This is the suspensor cell. Now the octant undergoes various transverse division to form a globular structure. The further one or two stages are after this stage. There are further more stages which results in the globular structure, and then afterwards a heart-shaped structure of the cotyledon is formed. The suspensor starts to reduce or degenerate. So there is no role of suspensor afterwards. This suspensor starts to degenerate or reduce. Then a structure, this type of structure is formed. These two are the cotyledons, just looking like two arms. These two are the cotyledons. This is the radicle. Now here, cotyledon, the portion which is above cotyledon forms the plumule. The portion which is below cotyledon forms the radicle. And just between this, the lower portion is the hypocotyle. And this one, the quadrant I have drawn. Just let me draw. the structure which is formed in the dicotyledons. Cotyledon, the portion above the cotyledon forms the plumule, then the shoot, and the portion which is below cotyledon forms the root or the radicle from the radicle. Then next to this is the monocot. Divisions or the development of monocot. Let's start with the zygote. Here the zygote divides into by transverse division into basal and apical cell. The upper portion is the basal, lower is the apical cell. Basal cell again divides by transverse division to form Ci and Cm. Again further more divisions takes place in this one to form the suspensor, which is denoted by N and M in case of monocot. Here next stage is the longitudinal division, which is similar to the stages up to the quadrant stage is similar with the dicotyledonous. Afterwards there is a change. So this stage there is a longitudinal division in the apical cell. Again the apical cell undergoes transverse division, but the cytoplasmic division is not present there, only the nuclear division. So 4 nuclei is present here, 4 nucleus. After so many divisions, the apical cell, this lower apical cell forms this globular type of structure, and the cell which these two are the suspensor cell. The cell which is in close contact with this globular portion is termed labeled as M. Now after more divisions, after further divisions, transverse division, the structure appears to be this one, where the N and N dash, which is the embryonic, pre-embryonic structures, now which are and they degenerate after the formation of cotyledon. Here the M is in M or the micropylor site is in close contact with the I dash or I. I dash forms the epicotyle and I forms the hypocotyle. This here I have not drawn all the cells in this portion. In these cells, these are not empty spaces. Cells are present. So just to make it simpler, there are 4 chamber-like structures with a deep ridge here, this type, and the cells are present in between. So empty spaces are not there. It is filled with differentiated, undifferentiated cells. Epicotyle. Now after this, this is the LS of mature embryo, monocot embryo, mature monocot embryo. Monocot embryo. In this structure, this one is a shoot apex. This heart-shaped structure, somehow it looks like appears to be like heart-shaped. So this structure is the shoot apex. It is covered by an undifferentiated mass. This mass is the undifferentiated mass, which is the coleoptile. It covers the shoot apex. After that, before this, the development of shoots or leaves, primordial leaves. So, and in the lower portion, the radicle is covered by the root cap, and again coleorhiza, which is the undifferentiated mass, to cover the radicle. Scutellum. In case of monocots, the cotyledons are termed as scutellum. Here. Now this is all about the development of embryo, stages of development. Once or twice, if you are drawing the diagrams, you can easily make it. You can easily understand the process. Next is parthenocarpy. Parthenocarpy is the development of embryo without fertilization, or development of seed without fertilization, fruit without fertilization. That without fertilization means if the there is failure of fertilization process and fruit formation takes place, if the pollination process or the fertilization is not proper, then all these conditions lead to the parthenocarpic development of fruit. And polyembryony is that in a regular cases, or in most of the cases, there is one embryo inside the seed. But if there is, or in the condition when there is more than one embryo, in a seed, then the condition is termed as polyembryony. Next is the apomixis. Apomixis is the condition where sexual reproduction is replaced by asexual mode, asexual reproduction. For example, in case of Asteraceae and grasses, graminiferous grasses, here Asteraceae grasses, there is flower formation and the seed formation is without fertilization or in the asexual manner, then the process is termed as apomixis. Next is the seed formation or seed. I am rubbing this part. Seed formation. In this, all of us know that ovary forms the fruit and ovule forms the seed. Seed formation, in the dicotyledonous seeds, there is a longitudinal ridge-like structure that this is termed as raphe. Just below this is a scar, and this scar is termed as hilum. At the end, at one end there is a minute pore, and this pore is the micropipe through which water enters, resulting in the swelling of the seeds and germination process. In the seeds there is two layers. Outer is the testa, inner is the tegmen. Inner tegmen, outer testa. In the monocotyledonous seeds, this is a epidermal layer between here the coleoptile and the plumule, lower portion the radicle covered with the coleorhiza. Here plumule covered by coleoptile. This one radicle and coleorhiza. Here a single layer is shown in this is the endosperm, and just at the outermost 2-3 layers of the endosperm forms the aileron layer. Aileron layer represented like this. This is a monocotyledonous seed. This is endosperm, aileron layer, outermost is the seed coat. In this aileron layer, it is formed by the outermost 2-3 layers of the endosperm. Aileron layer is rich in protein, while the endosperm is rich in starch, which forms the grain of the monocot seed, or the grain portion which is rich in endosperm, or the starch which is rich in starch, and aileron layer is rich in protein. This is about the structure of seed. Next is the fruit formation. Fruits are of two types, they are considered as true fruit or false fruit. Depending upon their mode of formation or their developmental process, fruit, false fruit and true fruit. If the development of fruit is only from ovary, then the fruit is considered as true fruit. While if any of the floral appendages involved along with ovary. Along with ovary. In the fruit formation, the resultant is the false fruit, or it is termed as the false fruit. Examples are strawberry, cashew nut, and apple. These are the fruit form process, true and false fruit. Then the layers, it is having the fruit have 3 layers, out coverings, outermost, pericarp, middle, mesocarp, and the inner one is the endocarp. Now this one, outermost pericarp is the outer covering, and which is hard, tough covering. In case of fruit, mesocarp is the portion which is eaten by or which is consumed. Now in case of dry fruits, pericarp is pericarp degenerates or dries up, while in case of Coccus nucifera, mesocarp and endocarp is in close proximity with the developing embryo. So all these things are related with the developmental process of monocot, dicot, and the seed and fruit formation. This chapter is complete today. In the next class, we'll start the genetics, and afterwards human reproduction. So first we'll start the genetics chapter in the next class. Thank you for today's session.